The next speaker is Pavel Sinitcyn, and he got his master degree in 2014 in Lomonosov Moscow State University. And then in the same years, he moved to Germany, working as a PhD student in Max Planck Institute of Biochemistry. He focused on the integration of next-generation sequencing data into a proteomics pipelines. And then today, he will talk about the next-generation sequencing in MaxQuant. So welcome, Pavel.
Thank you very much. I think that's all talks before we were like mostly about mass spectrometry, like about proteomics in general. But for sure we have some big elephant in our room, yeah? So, and this elephant is next generation sequencing. So basically, yeah, it's true. Like if you look at the flow of information and the addition of the degree of freedoms, going from DNA to RNA, you have many additional degree of freedoms like expression, splicing, modification, secondary structure. But even going on a protein, you're basically multiplying all these things, all these factors, on many another factors, like expression of proteins itself, interactions, cell localization, and post-translation modifications. Yeah, I mean, for sure, mass spectrometry and proteomics, it's really a huge field, and there's really like many things to uncover, but why not to use some advantages which we already have on the 2 floors above, on the DNA and RNA level, in order to improve our projects which we are doing on the protein level. And actually why not? There's also examples when proteomics can really help a lot to the 2 floors above. So, but what like next generation sequencing can do now? So starting from the chromatin IP and epigenetic marks and the chromatin structure, whole genome sequencing, exome sequencing, like mostly patient data, mutations, chromosome aberrations, DNA modifications, and going already on some expression level on genetic information realization information, like levels. mRNA sequencing with all things, interesting things there, like isoforms, mutations, expressions, secondary structure, even editing. And like starting from 2009, also ribosome profiling, which allows to see how translation is going on in the cells. So, and actually, ribosome profiling can do really many things, and especially it's interesting for us because it's it's basically the closest step which NGS can reach to the proteomics. Yeah, and one more thing, you can interrupt me, ask any questions. So whenever you want, just raise your hand. So, but yeah, and the main question, what actually can we do? Like, and one of the really the basic example is just mutation calling in the proteins. So the thing is that maybe you already know from previous presentations that, like for example, MaxQuant and many other softwares, proteomic softwares, we're working on a principle of constructing, first of all, database, and next, trying to find the best match of the experimental spectrum into this database. But the problem is, if you didn't include something to your database, you for sure will not find it. And for sure, it's true for mutations, which are non-semantic mutations. Non-synonymous mutations. And an idea here that let's kind of take NGS data, like patient-specific or for some cell line which is publicly available. There's basically many fragments, and there's some variation in certain position when you align the genome. And you can find out that this variation is actually changing amino acid. And it would be really nice to include it to database. And yeah, actually there's some story behind this slide. So in our institute we have seminars like where you're presenting your work, and I really try to make a lot of fun, like to play around one film, but it didn't work out. But okay, so but yeah, what I want to tell you, I want to tell you one example way it can be useful. So I think, in my case, for example, I saw many papers when people just, for example, taking some cancer cell line and making proteomics and transcriptomics or genomics for these cell lines, and basically finding some number of mutations and including this mutation to database and searching for it. But from another point of view, it's actually not cool, let's say. We can for sure find mutations on a protein level, but basically genomics has much higher power to find these variations. And what we should do, actually, we should go in some directions where proteomics and real power. And this real power, for example, can be really obvious in the case of immunopeptides. What is immunopeptides? So, to answer this question, we should ask another question: how do T cells recognize criminals? So basically, there's a really tricky system. Like every human cell expressing MHC1 complex, and this MHC1 complex presenting some random peptide length of 9 amino acids on the surface, and any T cells which are floating around can sense it, and something can happen wrong in this case. So what can happen wrong? We can have peptide with mutations, and when a T cell recognized as a not host peptide, it will sell some signal or start some signaling in order to kill this cell, because this cell generating some peptides which are unknown with some mutations, and better to kill it than lose all body. And the same about viruses, actually. So basically, viruses introduced to the cell, and all proteins which are synthesizing can be also randomly chopped and introduced, like exposed on the surface of the cell, and the immune system will recognize this peptide and kill it. So, and what's actually our idea? That's what can we do? We just can basically take some cancer cells from patient, for example, and remove, like wash out all peptides on the surface. We can do it in a more tricky way, but basically it's the simplest way to do this. And the next, basically, we can take tumors from a cohort of the patient, and wash out all peptides from the surface of the tissue of this patient. So it's in parallel to the whole exome sequencing or whole genome sequencing as you wish, or even actually transcriptomic. So you are sequencing tumor and sequencing also PBMC, basically the blood cells from the blood, in order to compare this data set from PBMC and tumor in order to find actually somatic mutations, basically mutations which are not happening in the blood, but somehow appeared in the tumor. So basically, probably these mutations which are specific, mutations which are specific for this tumor. And the idea here that we're just taking all this genomic data, integrating to the MaxQuant, basically taking all these mutations, non-synonymous mutation, and including to the database. And next, we are searching for these peptides. We are also searching for a normal reference peptide, but as well, on the top of this, we are searching for mutated peptides. And as a result, we are finding not so many, but we're finding, for example, like 5 peptides per patient with somatic mutations. And what's next? Of course, it should be immunogenicity assessment. Like we just have to check it's really actually there's some immune reaction on it. And in the future, probably, if everything would be fine, we can actually use these peptides as vaccination, like personal vaccination. Basically, we are going to train immune system of the patient in the body or out of body, and train against own cancer. So sounds nice, but there's many technical problems with these peptides, as you realize, because this is not tryptic peptides. And as a result, basically, we're flying bad. And we are 9 amino acids, not so long. But it's a super interesting topic from my point of view. It's worth to pursue. Yeah, you maybe saw, or not yet, but actually this is a patient where we have melanoma mutations. Melanoma cancer. And why exactly we choose melanoma for the first draft? Because melanoma, okay, this is a graph showing the different type of cancers, and basically number of somatic mutations somehow normalized. And you show that melanoma is basically top 1 of amount of the mutations which basically cancer accumulate. And that's why it's actually really cool to work with melanoma, because basically we're generating many non-synonymous mutations. As a result, we're generating many peptides with non-synonymous mutations. And basically, it's a good way to go. Yeah. So one more thing. That's actually, we are not it's not breakthrough, let's say. Breakthrough in the sense that people were doing this before as well, but without proteomics. So basically, we were taking the genomic data from tumor as well as from the blood, like as a reference. We were finding somatic mutations. And next, basically, we are including these mutations to the protein and going through this protein by window, like window of 9 amino acids, and trying to find all possible 9 MERs which has somatic mutations. And next, what we're going to do, we know MHC type of this patient. And next, we're going to predict affinity of this peptide, or possible 9 MERs, against this MHC, which is specific for this patient. And you see in this case, you don't need any proteomic at all. But there's one problem. This model, actually, which I predict in affinity, were working not so perfect. And for example, let's look at this case. There's a mutation in this gene, SYTL4, like some amino acids change in another amino acid. If you basically score it, if you calculate prediction, if you predict affinity of this peptide with these mutations, you'll find out some really small number. It just means that this peptide actually really has a huge affinity to MHC. And that's why the rank of this peptide, it's 18. But just think about this. We have 17 peptides before, which algorithm predicted as really high affinity peptides, but we don't see it in proteomics. Yeah, you can debate with me like why we don't see this in proteomics, because probably we're flying bad. We just didn't identify them. But if you have additional support from proteomics, that's this peptide actually really presented on the surface, it's really a big deal. And instead of checking all 18 peptides, we just, using mass spectrometry, we can come to this peptide and go directly with this. It's like additional support that this peptide actually presented on the surface. Yeah, it's really, there's actually another thing that's community who are developing these softwares, who are predicting affinity. Actually, we're now start changing a bit in the sense that we're trying to make machine learning based on the proteomics data. And actually, it's, I think it's going to be really a big breakthrough in a sense of the increasing of the sensitivity of these methods. Any questions? There's one question there.
So this is a bit more on the biology side. So these peptides that are presented by the antigen presenting cells in the melanoma cases.
No, I think we're speaking about MHC1. MHC1 presented on every cell.
On any
MHC2, yes. It's
okay,
actually
but so then these peptides, the mutated ones, are coming from the cancer cells, I suppose, right?
Yeah, yeah.
But then you did propose to use them as vaccines, or?
First of all, I'm not MD, but
no, but I mean, I was wondering what the rationale for using them to inject them into patients or something like this is.
Okay, my suggestion that's at least it can be used to improve our algorithm of prediction of affinity, like all these algorithms which are predicting affinity. It's at least. And actually, and of course, in the bright future, it can be a really nice idea for vaccine, because like it's just natural vaccine.
It's a protective effect, right?
Huh?
It's a protective effect. So the cells are being targeted for being killed by the T cells, and that's why they're presenting them. So it's not something that you want to try and.
But wait,
what I mean?
We agree that the cancer has certain type of have a certain set of mutations, and basically what we are going to do. And we didn't find such mutations in the blood, in the reference. So as a result, targeting these cells which are presenting this mutation on the surface, we are targeting cancer. So we're basically trying to destroy it by a natural way.
Yeah, I get your point. Thank you.
Yeah, but it's also probably, I don't know, in the near bright future, it's probably can be used with like blockage drugs, like checkpoint blockage drugs, and yeah. But I think it's really worth to pursue this this direction because it's supernatural. It's not like another more invasive methods to treat cancer. Yeah.
Answer to the question. I'm coming from the field, and immune system, by getting these peptides, would be stimulated and then kill cancer cells. So this is a way of neoepitope strategies that are coming into clinics. But we can also discuss in that break.
So thank you. So yeah. And actually, another idea for the poster, which was not suggested by me, it was like one flew over immunopeptidomics. And basically and now it can be nicely said that we made one flew over immunopeptidomics. It was not crazy, but it was fine. So next thing, how we can use genomics and proteomics together, and I think it's actually a very beautiful topic. It's allele-specific expression. And it's especially, like, of course, we started with something which is pretty obvious. Like, okay, do we know from which cell line this in which cell line it's happened? Like a triplicated of the chromosome 8? Okay, the famous cell line HLA. Yeah. So most of the genome actually it's triplicated. It's actually like horrible for experiment, but it's really nice for allele-specific expression. Because look at for example, this position on chromosome 8. There's some gene there. Honestly, I don't care right now which one exactly, but in certain position, we see that like there's a GAG. It's a reference sequence, a reference triplet, and in the HLA cell line, we also see the like reads fragments from NGS, which contains C in the last position of the amino acids, and this is actually non-synonymous mutation. And if you think about this, if you, for example, imagine that all 3 chromosomes expressing the same amount of the RNA and same amount of as all this RNA producing the same amount of the proteins with the same degradation properties. In this case, we will expect to see some ratio 1:2 on amount of the reads on RNA level. So basically, if you count all reads which contains reference nucleotide and divide on the amount of reads which contain alternative nucleotide, we will have some description of the what's the ratio of the expression of different RNAs. And we can basically do the same on the protein level. It's a bit tricky. And actually, we just can, for the first round, we can just sum up all intensities through the fractions, through the charge, through the modifications. It's not the best, but for the first moment, we can do it. Yeah, you can debate with me. Like, for example, that's peptides with this mutation, and if this mutation can behave completely different in mass spectrometry. And it's true. But let's just, first of all, try and see what's going to happen. And basically, if you plot like such a heat map, basically where you have a protein level, basically ratio log2 of intensity reference to intensity alternative, and from another side, you plot it for RNA, and this is just log reference and log alternative. So basically, we see that we have some spot in near of the dot -1, -1. So it just basically means that, okay, we have triplicate here. It just means that we have ratio 1:2. So we have 1 chromosome with reference and 2 chromosomes with alternative. And basically, it's both the same on protein level, on RNA level. Cool. We can go a bit further. We have not even more. We have foplication. I don't know how to say. And the thing is that we can just take this data for expression and just sum up with previous graph. And we will have some really cloud, some nebula around of the plot, dot 0,0. What does it mean? It just means that the 2 chromosomes contains reference and 2 chromosomes contains alternative. And yeah, you may see another graph like here, 2, 3, 4 copy number. And actually, this data were retrieved from whole genome sequencing and just calling and finding how many how many chromosomes we have. And more thing, we can just take all our mutations which we found in proteomics, but you see it's really important. We should find the mutated peptide and non-mutated peptide. It's actually not so frequently happened, but we took HeLa and we took really deep data. And if you take proteomics data and RNA data together, you will be a bit sad. So why? Because look at the copy number 3. For example, for this position, we know that's why I presented the copy number 3 in the cells, in the HeLa cell line. And basically, we see two clusters, cluster near the point -1, -1. So basically, it's how expected. It just basically means that in one chromosome, we have reference, and the two of the rest of the chromosome, we have alternative. And we can see another cluster in near the point 1 and 1. So basically, reverse situation, when you have two chromosomes with a reference. But you see the standard deviation of these clusters on a proteomic level. It's sad. It's really huge. Yeah, what can we do with this? Probably we can improve our protein level estimations a bit better. Maybe we can use a DAA, or it's nature. Maybe we should select some peptides which has the same. Maybe we should select just such pairs of the non-synonymous mutations where both amino acids have the same physical chemical properties. We are working on it. But in general, it's actually in a bright future, near bright future. What we wish to see, imagine we have a dot around this place, like basically -1, 0. What does it mean? It just means that we have a copy number 3, and on a level of protein, we have a ratio 1:1, and on a level of RNA, we have a ratio 1:2. And it basically means that something going on with silencing of certain part of genome, sorry, in this case, it does mean silencing of part of genome. In this case, it means that probably this protein degrading in a certain way. Probably this mutation making protein more degradable. It's actually pretty interesting. So, and basically, you have USB sticks with you, and you have actually just one small comment. This is a special version of MaxQuant, which even not available publicly. It's like 1.6 point something A. It's mean anniversary. So basically there's an Easter egg there. It would be really funny if you find it. Yeah, there's a few questions there. Yeah.
Hi, I have a question regarding the protein quantification. Oh, this is really big echo here. We're usually using IBAGs to compare protein expression between proteins in one sample. Does that work with proteins with only a single mutation, where you basically only can distinguish them with one unique peptide, or?
Yeah, the problem here that we are not speaking about protein level. Like basically, let's say we have a big protein and one mutation in it, and basically 90% of the peptides from this protein are going to be from alternative chromosome and reference chromosome. Yeah, and in this case, we are really making most horrible quantization between peptides.
Okay,
so
thanks.
Yeah, it's also adding headache. Yep. So the thing is that everything is accessible in MaxQuant. You just have to go to the tools and go to variation extraction. You'll see such a window where you should specify files, and these files, it's NGS alignment files, like in format of BAM, which is basically standard right now in NGS. And you also should specify on the global parameters, you should specify genome FASTA and genome annotation GTF. So what's a genome FASTA? It's just basically a sequence of DNA, and genome annotation is basically mapping of different genes, different structures in a genome. And what we're going to have at the end, I'm going to play in between two screens. I guess it's going to be fine. So as a result, you will receive such such set of the files in a folder named final. Yeah, and about final actually, it's a really kind of big point. So yeah, a few friends actually point me on it. That's never ever call the documents final. But as you see, I'm doing this oppositely, and I'm naming even like folder final. So and you can open this file, and it looks like a normal FASTA file, except this thing. You see this kind of ID of the protein, and the next through the tab, there's a ID of mutation, amino acid before, position, and amino acid after. There are even cases when you have many mutations. For example, this one, you see. There's many mutations, and actually what's MaxQuant doing when you have many mutations in really close by to each other, MaxQuant taking all possible combinatorics of these mutations, which actually totally makes sense. Okay, and okay, you created this FASTA file, what should you do next? And you should like make MaxQuant aware about this, this FASTA file. And if you like go to USB sticks, basically, I will just demonstrate now. Go to MaxQuant, to this Easter egg MaxQuant. You will be able to describe the rule for the MaxQuant how actually find out these mutations in the same way as to find out identifier, and the same way as to find the description. So where you should do it? You should do it in the global parameters. Actually, I'm the first person who is showing MaxQuant, some practice. Sorry. So and basically you should add this FASTA file first. Where did I put this? Sorry for this. I put it in the folder final. It's exactly here. So and you see, that actually exists. Identifier rule such, description is such, but variation rule is empty. And we can add a variation rule just like clicking on this button. Oh my God, this is, okay, it's here. So basically it's just some help for you. You can just select some pretty classical regular expression to find this mutation and check OK. And honestly, after answering for many questions in a Google group about regular expression introduction, I would really ask you all the time check the parse rule which you used. Like for example, in this case, we will see that variation actually were found out, and we're creating this small set of variations. Really nice, and making this thing. And this is a bit redundant, but soon we're going to change it. But now it's obligatory. You should change and read from FASTA file and run MaxQuant with your raw files and find mutations, like peptides with certain mutations. Oh my. Okay, so one more classical example of usage of the NGS for the good of the mass spectrometer, it's gene expression or transcription expression analysis. So how it looks like from perspective of the genomic person, basically you have a machine standing somewhere, it's machine generating FASTQ file, and you're working a lot with this FASTQ file, making quality control, etc. And basically at the final step, you're aligning them to genome. As a result, you have a standard BAM file, and this BAM file, which is basically just alignment, should be somehow converted to gene expression table. So yeah, but the fact that currently there's a huge amount of databases like INA, Geo, ENCODE, Ensemble, which actually providing you many BAM files for tissues, for special cases, for cell lines, most frequently used cell lines, etc., etc., etc. And you can just load this BAM files and do whatever you want with them. So, and what was the idea here? That's why not to just make shortcut? So basically, many people can just load these BAM files, and we want to convert these BAM files to gene expression table for certain cell line, for certain species, etc. And why not to integrate this to Perseus and make it basically user-friendly and simple? And this was actually our goal, and we made it. So basically, if you open Perseus, you'll see such a window like next generation sequence data upload, such activity. And what you should do, you just have to add your files and specify many parameters. One parameter which is also obligatory important is genome annotation. You just have to tell to Perseus actually where the genes are placed. Okay, you have alignment, but you don't have any information about genes in this alignment. And yeah, so what I'm going to do right now, I'm going just to walk through these parameters. Like in one first parameter, it's strand specificity. Probably many of you are familiar with this, but I will just come to the ground floor. So strand specificity, it just means that we have our fragments from genome, and we have our fragments for RNA, and by nature, RNA is basically transcribed from one side of the DNA. And the thing is that the first experiments to make RNA sequencing, we were not kind of caring a lot about strand specificity. So basically, all fragments, let's say our genes directed to the right, but all our fragments were directed in a random way, 50% on one way, 50% another way. And this was just simple and it just was robust. But with, like if you want, for example, to learn something about antisense transcription, it's not way to go. But further people developed another way to process these samples into the strand specific when you preserve direction of the fragments, or like another way, just antisense, like also stranded, but antisense. Yeah. Okay. So there's many more parameters here, and actually, if you open Perseus, if you just move your mouse on the name, you actually will see help about this parameter, what this parameter actually means. And one also important parameter is, for example, feature type name. As you see, is by default exons. So what does it mean? Actually, if you think about this, in the case of translatomics, in the case of ribosome profiling, we have fragments just from the coding sequences. So in this case, doesn't make any sense to calculate the coverage of the 5 UTR and 3 UTRs. And as a result, you can just specify, in the case when you're analyzing ribosome profiling data, you just have to specify here CDS, sorry, as a feature type name. But for the common RNA-seq data, exon, it's the way to go. If any questions, please ask. So, sorry. So the thing is that we surprisingly found out, okay, this is not surprising. That's basically the Perseus implementation allows you to use several threads. And actually, these threads, it's not recommended to use more threads than number of files for now. So, and it's true, like with increasing of the size of the files or size of the amount of the fragments in files, you see that you are increasing time of the calculation. But with using more threads, you can reduce this time. Not drastically, but you can reduce. So, and the thing is that we are not first in this place, for sure. I mean, this is kind of this topic which was raised from a long time ago. And there's even like some Python framework, which actually is super popular, which is used for just calculation of the amount of the fragments per gene. And it's named HTSeq. And if you compare, actually, if you take some data samples from a geo database, like replicate 1, replicate 2 from a certain cell line, and with such notation, you'll see that's kind of, if you look from the Perseus side, you see that's correlations when you have, we actually compared directly Perseus and HTSeq count, like replicate 1, replicate 2, replicate 1, replicate 2, and we saw that, for example, if you take this case, there's a like correlation 1 between HTSeq count and the Perseus. Basically, we're generating the same results. And of course, we have some variation in the case of when we're comparing 2 replicates. And the thing is that another really game changer, that's it's really fast. If you compare compare Perseus and HTSeq count, Perseus just really much faster. And actually, why it's game changer? Because it allows like for common users just to take like some BAM files and analyze it in a different ways, how we want to do, and and play with data. It's actually a key thing about Perseus, and that's it allows you to play painlessly play with data. So, what can you do with this? One thing, which is actually, from my point of view, just beautiful, but not so much biology under this. So you can compare 3 omics. You can take translitomics, you can transcriptomics, and proteomics. And what can we see from this picture? So we can see that the weakest correlation going to be between translitomics and proteomics, which is basically really small. And there's many debates. Why is it happening? Let's not go to this direction. But what's interesting, the next highest correlation is correlation between translitomics and proteomics. And actually, it's basically really nice because it just means that it actually was written even in original paper of which presented ribosome profiling. That's actually translitomics, ribosome profiling. It's really the closest proximity for expression of the proteins. And it's really no surprise that we see a higher correlation between translitomics and proteomics, higher when transcriptomics and proteomics, because between them there's a translation which introduces more parameters for like more degree of freedoms. And the highest concentration, highest correlation going to be between transcriptomics and translitomics, which is basically the same technologies, less noise, and nice. So cool, we can compare 3 omics, but the thing is that sometimes it's really worth to just compare 2 omics, in certain cases. And this was a specific case, which actually I really liked a lot. So basically, EMC2 protein, it's a chaperone, which basically shaping the proteins in a certain way. So, and the thing is that you can kill this protein and see which proteins actually affected. And what is really nice, you can just plot the ratio of protein abundance when you kill this protein and control against translation. You can check out, again, when you kill EMC2 and control. So translation means ribosome profiling. And what is really nice that we see some cloud of the dots, basically, which are not changing translation, but we are changing a proteomic level. So basically, even more where the proteomic level is decreased. So basically, it's actually what we're expecting from the influence of chaperone. If you kill chaperone, we expect that translation in a small time scale, translation should not change, but the biggest effect going to be on proteomics level. And we actually see this. That's many proteins who are actually dependent from EMC2, we are not properly folded, as a result, were degraded. Nice. So one more thing, which you can do really fast, I mean 41 minutes. I even have lunch, I think, more this time in general. So you can load these files, which are basically 100 GB from ENCODE project. This is 1, 2, 3, 6 cell lines, and with 4 replicates, even more. And basically, you can throw all these things into the Perseus and analyze it. And this analysis will take just almost 1 hour. And you will get, you can plot PCA, and you can see that's basically it's how expected. Yes, so basically cell lines grouped together. That's basically how expected, and how it's really nice. And actually, I don't know for sure, but the thing is that these cell lines were related in the origination of the tissue origination. And these things can be done really fast. So yeah, I would actually, with my home network, I think it will take more time to load it when to actually process it. So really nice. Yeah, and with this, if you have any, okay, let's first of all say acknowledgement. So thank you very much for our Cox group and Matthias Mann group for any support. And thank you really a lot for support of our collaborators, which without whom it would be not possible, all these projects. And yeah, and thank you very much. And if any questions, please ask.
Are there any questions? We have some time for the questions. Yeah.
Yeah, hello. I think it's really cool to be able to do next generation sequencing in MaxQuant. And one thing I thought about, I know that proteomics is a hot topic. It would be really cool if you could generate Or you could identify, for instance, mRNA sequences, and then you could translate that into protein sequences, and you could create some kind of customized protein database that you then can search your MS data against.
Okay, so one thing I would say current annotation is really good. So it's really happening very infrequently that you're finding something new. And in this sense, probably it's not so much sense, but there's actually another thing which actually we are doing. Right now it's in the progress. Idea that we can just basically take RNA-seq data, assemble it, and make free-frame translation. I mean, why free-frame, not 6-frame? Because we have a direction, and we for sure know what's going on there. We can like put some limits, number of minimum amount of amino acids, like starting from methionine or not, and taking all these things into account, you can make free-frame translation from your raw data, and basically create faster file which basically originated from the cell line, etc., etc., etc. Which is basically really nice. But yeah, I mean, I would say it's in the progress, and we are thinking how like put it in the best way, because like this assembling step, it's not so simple, but we will find out some solution, simple solution. But we are doing this right now.
Thanks.
I think there's actually the thing which I didn't mention, but was also super important, and actually previous time I asked really a lot about this. So if you think about this, like all what I was doing here, maybe I tricked you without saying this. We are doing here gene expression table. It's actually not transcript-specific table, which is actually important. So why are we doing this exactly in a gene expression level? Because if you want to go on transcription expression level, you actually, because of the many, because basically all isoforms of transcripts created a graph, it's actually becoming a bit tricky to make a quantification on the level of the, even on level on transcriptomics. That's why there's like many papers which are suggesting different models, different assumptions, and based on these assumptions, we're making different quantification models for isoform-specific quantification. So the thing is that in the first round, we decided to make as simple as possible. So basically, all fragments which are generated from certain read doesn't depend like with these fragments coming from splicing junction or somewhere else. We are merging them in one definition and one gene. So I would say again that's it's actually not so simple topic like transcription isoform quantification, especially on the level of proteomics when you don't have 100% coverage in most cases. But let's say we are right now working on it for the high coverage data sample. And we're just trying to explore this direction of the movement. And let's see.
Okay, I have a question as well. If we compare, that's why the result that you show, if we compare different omics, and then the correlation coefficient is very low. But did you also compare only mutations? Because in your previous results also show that actually the mutations works quite well. I mean, better than compare whole genomes. Do you also have some like correlation coefficient number in mind?
I'm sorry, correlation. I not quite follow. I mean, correlation coefficients between what and what?
Only mutations of the RNA sequencing data and then the proteomics data.
But it's basically binary correlation, you want to say? Like this mutation is presented on RNA level and on a genome level, or what you're asking?
In your previous result, the result that you show, we can see the genes. For example, here it's like the RNA, there's the mutations.
Yep.
And then in the protein, they also have the mutations appears. Do you have something like overlaps between like the RNA sequencing data and proteomics data? Both can detect the same mutations.
Yeah, but basically the whole analysis is based on exactly the subset. The subset,
yeah.
Actually it's really important, subset of the mutations which can be detected in both. It's even more, it can be detected both on RNA level, on a protein level. I would say it should be identified on RNA level. That's why we're finding it in a protein level. Mm-hmm. And one more thing, we should find it on the reference. We should find it. We should find both. like protein peptide from the reference with reference variation and with alternative variation, basically from both chromosomes. Yeah, maybe you want to ask me a question like, what about the transcripts, which are actually very low abundant, but the proteins are not very well degradable?
Yeah, exactly.
Which is a good point. But I think actually in this study, we were taking mutations from genome side. We're not taking from transcriptomic side. And yeah, the result basically how it is. Yeah, probably it makes sense to plot some Venn diagram to find out, like, in a binary way, which mutations we found on RNA level and which we not, and compare with proteomic level. It's actually a super interesting topic.
Right.
I completely agree. And plotted with half-life of both RNA and protein.
Okay, here on the right side.
Hi, Pavel. Thanks for the talk. Did I understand correctly that you took the HD-Seq idea and implemented it yourself, but much faster?
Yep.
Congratulations. That's cool. Do you make it available in like batch processing mode that you could use it in a standardized gene expression thing, or is that accessible through the MaxQuant?
I'm sorry, I don't understand what you mean batch effect.
In command line fashion, that you can streamline it on a Linux server for high throughput gene expression quantification. I'm
afraid for now, no. I'm sorry, but this is totally makes sense because, like, basically, whole genome, yeah, is for sure, you know, like almost all genomic field working in Linux.
Do you
not
make
yet
the source code available on GitHub?
We move it there. Okay. I'm sorry. I mean, it was just under development. So
yeah, it's not urgent for me. It's okay.
No, but this is important. Yeah. I am pretty for this, and we kind of actually during MaxQuant summer school we are doing to-do list, and this is going to be fourth point.
Yeah. Thanks.
Thank you. Any more questions?
Yes, here.
So I think in the next slide, yeah, exactly. Do you think you're going to follow up maybe with some experiment like the protein degradation that you were suggesting that could have an effect on the peptide that give rise to this standard deviation?
Yeah, I would rather, like, honestly, it would be nice to find out, first of all, the case when we for sure know that certain mutation is making protein degradable much more. I think it would be a good starting point, because for now we don't know actually different properties. We have basically sum of two effects: some of the noise, some of the different liability of the peptides, and the sum of something which we don't know. And as a result, it's very hard to fish out the effect of the biology. Sorry to say, but we are thinking how to do this. But I think the good way to go first, it's find out some biological case when it's takes place. Yeah, we are considering to follow it for sure. So I think.
Okay, there's no questions. Let's thank Pavel.